The next speaker is Professor Shai Shen-Orr from Technion, and he will tell us about immune system aging and inflammation.
Thanks everyone, and apologies on missing the first part of the day. We need a microphone for people. What I'm going to tell you today is a study started actually in my postdoc at Stanford as an ongoing longitudinal study looking at developing immune monitoring and specifically looking at changes with age. The focus is of this it started as a kind of discovery-driven science, but I think I could say ignorance-driven science. Let's see what changes, and we can measure it at a high resolution. But slowly it's been evolving into trying to figure out this paradox where in aging you have both a condition in which there's increased cell stimulation and increased pro-inflammation, which is relatively well known and established, and low cellular responses or ultracellular responses, as in the two I would say hallmarks of aging, and yet you wouldn't necessarily expect that to be the case at least in a dish in an acute condition. And most of what I'm going to show you today is going to be data from techniques that I'm not sure if everybody knows, so I'm kind of going to go over a basic slide of, and this is a technique called phosphoflow, originally developed in Gary Nolan's lab at Stanford. And what it is essentially doing intracellular staining, originally with flow cytometer, and we now do this in the lab with mass cytometer, where what you do is you take immune cells, you fix perm and use antibodies for specific epitopes of, say, phosphorylated transcription factors, and look at single cells by flow in unstimulated condition and in stimulation. So here in this particular slide, you can see you can look at cells simultaneously at a cell surface marker and identify cell types. It's kind of more standard, and then take and stimulate the cells, and I also have a marker that's intracellular for, say, transcription factor, and you can see then that a subset of the cells are responding to the stimulation. So this is a very quantitative way of looking at a single cell level at populations and signaling responses in cells. And what we did is we applied this to a cohort of individuals. Originally a pilot study that started with 29 people and has now been going for, we have 4 years of data and now collecting the fifth, where it's been expanded to 89 people followed over time. PhosphoFlow is just one of many assays that are being run on these folks. When we looked at that data, the first interesting thing was to see what huge variation we saw in responses, particularly in aging, in the age cohort. So you can see here the data of one of such stimulations. I'll show you the full picture in a second. But here we're looking at essentially CD4 negative T cells or CD8 positive negative selected that are being stimulated with IL-6, and we're looking at the full change response of p-STAT3, transcription factor that's it's high use in immune cells. And the data on the x-axis you see age, but on the y is the z-score, the normalized data of the population. You can see that essentially the young individuals are relatively high and also are relatively homogeneous, where there's a very big variation with age. And this particular assay that's just one, but in fact we've run this in 72 different combinations, looking at 4 different cell types. So you can see them B cells, CD4 T cells, CD8 T cells, monocytes, stimulated with 6 different cytokines, interferon alpha, gamma, 6, 7, IL-10, 6, 7, IL-10, IL-21, and looking at STAT1, 3, and 5 phosphorylation simultaneously in the same cell. So we can look at p-STAT1, 3, and 5 simultaneously and all these markers together in this heat map, every row of which is a combination of a cell type stimulated with a given cytokine and looking at the full change response of a given transcription factor with individuals ordered by age. This is the first pilot data from the 29 different people. Here showing you the full change response, so how much is this changed pre-pro stimulations, and here's the baseline unstimulated data. And every time you see a red spot here, this is essentially due to the fact that we found a significant difference with age, and in, of the 72 different assays here, 39 we could see were reduced with age. And you can see also on the other hand that, and we'll get to this in a second, that the baseline in many cases was actually increased with age. And an interesting point is that this is happening systemically in different cell types and combinations. And so the first thing that happens when you see such data is okay, well, I chose a dose for the stimulation, but maybe the elderly, we know they have high levels of inflammation. Maybe they're just used to a certain dose. Maybe their cells need a bigger boost to actually be able to respond. And so we did a whole dose response assay of the 72 different conditions in 4 different individuals, and you're seeing here just of the CD8 T cells, stimulated IL-6, fold change of p-STAT3, and looking here, you can see the dose response curve, and we can draw a fold change to concentration, and this is a very typical plot from what we get. Essentially, the young are able to continuously respond irrespective of the dose, and reach a saturation in the end. The old reach a saturation and are showing reduced response irrespective of what the level of the stimuli is. And this is systemic, so you kind of could have picked this up from the heat map. There are certain individuals who are poorly responding irrespective of what cell type you're looking at, generally, or what STAT protein you're looking at the fold change, or which transcription, or which stimulation you're using. And so, and I should say there's longitudinal data, so we can look at this across the different years and look at individuals, the population, do we pick up these differences? So shown here are 4 years of data. We have some issues with the 4th year, but every row here is a year, from year 1 to, so year 1 had 29 people, and then we're talking about 89 individuals. And every time you see a yellow, that's essentially, it shows you the false discovery rate. So the percent chance that this is an error, and this would be falsely discovered, and essentially across 3 years, in a false discovery rate of 0.2, we can pick up 13 stimulations that are consistently being downregulated in the elderly. And then there's less, there's more if we look at particular combinations. And this is at a population level, but you can ask how much is an individual stable. So shown here is one such example where we can look at, we're looking here at CD8 T cells stimulated with IL-6. And how much does p-STAT1, the fold change of p-STAT1 when we stimulate it. And you can see from year 1 to year 2 that generally individuals are stable in their response. The gray here are the old individuals, the white is are the young individuals. We had a group of 18 to 30 years old. And generally a person coming in, walking into your door and having poor CD8 T cell responses on 1 year, following year, it'd also be non-responsive generally. And so what drives this? So I mentioned I have a huge amount of data on these people with gene expression. We counted, we phenotype their cells, we know the different subsets of cell subsets that they have, serum cytokines in them, and so forth. You can take all of this information and start looking for correlations in the data. So just to make this a bit simpler, we sum up by different cell types, so we can look at, we're saying, okay, forget which stimulation it is or which transcription factor. What are the, can I sum up all the fold change responses in CD4s and CD8s and monocytes and so forth, and look for things that are significantly correlated with them. And here's the kind of the top of the list, and what you can see is at the top of the list are baseline levels of the different transcription factors. So the abundance of the phosphoprotein before stimulation, and the frequency of different naive memory cell subsets. And then there's things like in particular cell subsets, like in CD8 T cells. There's IL-6-ST, the receptor, and so forth. So to put this in a kind of a more cartoon-like figure of this relatively complex phenotype, what can drive this? I sat and thought about this for a while, and I came up with 5 different models. And shown here, the basic idea here in this figure is think about this as being your baseline level. The top would be your fold change, the maximum available total protein, and the arrow is your fold change response from baseline in young and old individuals. And then the flash little thing is the amount of stimulation you get. So one thing that could drive what I've shown you here, which is essentially a huge difference in how individuals, young and old, respond to stimulation and variation within old individuals as well, is just if naive and memory cells actually respond differently to stimulations. So it's actually well known that older individuals have more, say, memory T cells. And so if memory T cells respond poorly, they have reduced fold change responses, then maybe the total sum that I'm measuring of the immune system would be reduced. So that would be one model. The other model would be adaptation, which essentially says the old need a higher dose to be able to respond. And I kind of showed you the experiment already that this doesn't happen. The elderly don't respond irrespective of what the adaptation is. The stimuli, there could be a resistance defect, so something in these cells where the pathway isn't functioning properly. And so the elderly are not able to respond irrespective of the stimulation, or this idea of the baseline being up. So if the baseline is up, maybe I can't reach that, like I max out on the total protein, I'm just not able to make the same fold change response, or what we call the double whammy, combination of a few of these. So I can take all the data that we have and we can build a computational model to try and figure out for each one of different stimulations where they fall. And we did this with the dose, using the dose response data as well, and came up with this idea that really we expect many of the differences to be just due to how much you have naive and memory cells. So every person has their own little ratio, which changes over time, and memory will increase. And then the more memory you'd have, we expect to have reduced responses for some of those stimulations. We predicted, and then also we saw in the measurements that there wouldn't be any adaptation. Monocytes in particular we thought would have resistance defects, and then the rest would have either increased baselines or increased baselines and something wrong with the pathway, which is what we saw with IL-6 and IL-6 receptors that I kind of showed earlier. And so we took, from 89 individuals, we sorted naive and memory cells, repeated the phosphoflow assay, looked for differences within an individual between how much are they responding and fold change in a naive cell and memory cells, are they the same? And you can see here in these box plots are young and old individuals, the red and cyan, and 0 is if your cells are responding the same. Because these are generally cells do not, naive and memory cells within a person don't respond the same to the stimulation. And in fact, in most cases, the naive cells are responding better than the memory. And this effect, which I said, an individual's response may be just due to naive and memory fluctuations, is actually also based on the level of baseline. So what we noticed is that for the T cells, where we had the data, in many cases, naive and memory cells, in all the cases, they don't have equal levels of baseline. And in particular, where we noted that the poorest responses in the elderly, the baselines of the constituent factors were elevated. The phosphorylated abundance was elevated at baseline. And so even in memory cells, we think that what's happening is that we can't make this fold change response. So here's kind of an idea of what would drive this. We're looking essentially at the JAK-STAT pathway, and so STAT 1, 3, and 5. And a few years ago, Beckman and Al published a really nice mathematical model of the pathway and showing how, essentially it was a paper showing how this pathway is robust to stimulation. So no matter what the concentration of the stimuli is, the pathway is going to respond the same. You're going to get stimulated at time point 0, and then an increase in the phosphorylated protein level, so the fold change, and then a reduction to a steady state. And this was essentially a single shot of stimulation. And this not being an aging model, so everything here is kind of relatively quick time span, we said, well, what would happen if we actually gave in this model a chronic level of stimulation? And the motivation is that whereas the pathway may be robust, what I keep hearing when I talk to physicians is chronically high levels of things. So IL-6 levels don't matter if they're high in our predictors of aging sporadically. It's an issue if you chronically have high levels of IL-6. And so what would happen in this model if I actually gave the stimuli for a long period of time? And what we predict would happen is that the fold change would be made properly the first time, but then the steady state, the amount of constituent factor that you have after stimulation, when the stimulation is always present, the steady state goes up. So you're going to have baseline levels of the constituent factor being higher just for the fact that there's more, that the stimuli is always there. And this would even be the case worse if the stimuli goes up in time. as it is predicted to happen, measured to seem to be happening in aging. So this is the mathematical model. What happens with the data? So we can look at these people. 89 people, we're looking just at the, so 60 people, just the old people here, and we're looking for correlations. What the prediction is that the higher your baselines, the more you're going to have pro-inflammatory proteins. And so specifically, we looked at inflammation both with CRP measurements and IL-6, and you can see there's a weak but significant elevation. And in particular, if you take this now and divide the individuals based on their clinical parameters for CRP and IL-6, so CRP below or 1 or above 3 and IL-6 in the first or third quadrant, you'll see that there's significant differences in CRP levels and IL-6 in the sum of the baselines across your different immune cells. So essentially, what this would suggest is, and shown here is this is for the CRP, the p-value is 0.01, and for IL-6, or the tandem of saying you have both IL-6 and CRP together, the Fisher's combined probability test is very significant. And so the basic idea is that somehow your baseline levels of your proteins in cells increase over time, and that this would be detected at a clinical level with increased inflammation. And then there's an interesting phenotype. We noted that we took these 60 old adults and had a very thorough cardiovascular assessment of them using a bunch of different techniques, which essentially I've summarized here. And that is, we've taken essentially looking at like the sum of the plaques and atherosclerosis and so forth, individuals who have high cardiovascular risk within our what is generally a healthy cohort with no kind of apparent disease, have reduced responses of their immune cells with age. The p-value is below about 0.05, but I think given long story here is still an interesting piece. And to kind of summarize all of this together, it leads us to this model. And so the basic idea is that we have some kind of chronic condition. In particular, some of the things we've noted is that our poor responders were CMV seropositive, which would be one such condition, but could be many other different things. And that would essentially, over time, increase your pro-inflammatory environment, which we know, and generally in aging, goes up. Based on the mathematical model and our data, what we see is that the baseline of the T-stats goes up. Your full change response of the cells to the stimulations will then go down over time, and the cellular output, as we know, kind of as a hallmark of aging, is reduced or altered. And we think that this would eventually lead to kind of reduced immune responses, and particularly saw the cardiovascular data. That's it. Just to acknowledge folks in my lab involved in this is Irina Sorvetsky, my lab manager, and Val Barker, who's the cardiovascular work, and folks at Stanford where I started this work, Mark and Tool, my postdoc mentors, David Furman and Brian Kidd, who've been close collaborators, and Francois is the clinician with cardiovascular data on these folks. Thank you. Questions?
So when you see that when you have a smaller, lower response in people with high risk, cardiovascular risk, do you think that that's also these people have higher, and it's also from the previous results that you've shown, these people have higher cytokine levels in the immune?
Yeah. So they have, so the whole picture they tried to draw, I didn't show that in one slide, but it's all tightly linked. You have high levels of pro-inflammation. Those pro-inflammatory levels lead to reduced responses. I should say these people also have exhausted, kind of increased levels of exhausted T cells, both CD4 and CD8, so CD28 negative T cells, and then also a clinical phenotype. We've even noted we've done influenza vaccination responses on them, but we couldn't reproduce this over the 3 years. But at least in over the first, like, we saw this very strongly in one data set, in one year, and the second, a bit lower, we saw that they were, when you give them an influenza vaccine, they respond, but the titer doesn't hold over time. So they drop down faster. So it's all one phenotype. It's essentially an immune state, if you think. I sometimes say to the, I speak about this, I say, look at the people in this audience, we each have our own cell frequencies, we each, we think about the immune system as being all the same, but really it's a signature of everything that you've passed, and in the end, we end up in aging getting these very different states of the immune system.
One follow-up question. So now if you treat these people with anti-inflammatory drug, aspirin, right, would this change if you treat them?
So an interesting question. I don't know. So we're actually starting a desensitization study for allergy based on this idea where in an allergy they've already also noted increased baselines. It's noted both in allergy and asthma, and which is essentially could be reduced by different drugs. And so it's certainly an interesting direction.
I have a question. You said that for the most part, most of them behave the same between the years of the study. I was wondering, between year 1 and year 2, year 2 and year 3, the trends remain the same.
It depends on the assay. So some of them, particularly the CD8 cells, are relatively stable.
So I was wondering if you had enough cases where things have changed, and whether or not, because those people that got to this point had to change. So I'm wondering if the change is dramatic or is it?
We don't really have enough data on it. Well, we've tried doing that. It's a very interesting question. We've been pulling data together. What we are noticing, and again, with some reserve, is that when we see a young person get worse, it's temporarily, and when we pick it up again, when we look at them again, they would look fine again. With an older person, the chances of seeing them be a poor responder from 1 year to another are higher. The chance of being stable as an old person are higher than they are as a young, in particular, or being worse, right?
I'm just wondering if it's a slope, if it's a slope or if there's
We are wondering as well, but we need more data. To what extent would this approach be clinically feasible? Yeah. So I mean, I think the first question, so there's two pieces to this, right? A is treatment, and second would be diagnostics. Diagnostics are easier, and certainly one direction we're going. So we're collecting more and more data on these, and we're doing this in multiple different types of diseases. We think generally the approach here is that the immune system and monitoring the immune system is way closer in terms of when you think about personalized medicine. It's a much closer and much easier interpretable phenotype than to start with SNP data or kind of markers like gene expression. And so that's the approach. We still obviously have a lot of work ahead of us. I think there, certainly this idea of exhausted T cells, which I'm going to link to senescence, as Valerie was saying, that in itself appears over and over in a bunch of different diseases. We're looking at this in cancer now. We're looking at this in other phenotypes. I think this will.
The next speaker is Sivan Kondi from Bar-Ilan University. And he'll tell us about IGF-1